Yeah, welcome everyone. Just want to start off with a quick introduction. So my name is Peter Fowler. When I'm talking to beekeepers, I like to remind everyone first and foremost that I am not a beekeeper. I'm a veterinarian studying honeybee diseases. A little bit of background about myself. So originally from the Northwest, I did my undergrad at University of Washington in Seattle. Moved out to Michigan in 2016 to go to vet school. After vet school, I graduated in 2020. Kind of during the process, I became interested in public health, One Health, and disease ecology, which is kind of looking at systems in which diseases function. After vet school, I continued on at MSU for a PhD. I did a little bit of work with ticks and a little bit of work in poultry before landing in honeybees. So since 2021, I've been working with Dr. Meghan Milbrath, who will introduce herself in a second here, studying European Foulbrood. And I'm excited to talk everyone's ear off about European Foulbrood today. And most importantly, not a beekeeper. So any advice you get management wise about honeybee colonies you may glean from my part of this, I would take with a grain of salt, but I do hope to someday be one.
And then I'm the other half of tonight's presentation. So I'm Meghan Milbrath. I'm originally from Wisconsin and I did biology at St. Olaf and then a master's in public health from Tulane University. And then I moved to Michigan in the early 2000s to get a PhD in environmental health sciences and epidemiology, which is in the School of Public Health. So all of my background is really in infectious disease transmission and identifying risk models. So I started in human diseases and then I switched to studying bees academically for my postdoc, which I did at Michigan State University with Zachary Huang, looking at nosema co-infections. And then I did another rotation as a researcher at Swedish Agricultural University. And I am a beekeeper, but that doesn't mean that I'm going to be able to solve all of the issues. So I started keeping bees as a hobby when I was a kid in the 90s in Wisconsin with my dad. And then I've done bees as a business, so selling queens and honey production since about 2010. And so I do bees at work and bees at home and bees all the time right now.
European Foulbrood. I like to start off with this kind of side-by-side comparison. So on the left, we have an illustration from the UK of a diseased brood frame from 1885. And then on the right, we have almost exactly the same frame photographed from Switzerland 2015. So it's 130 years have lapsed between these two, and we still have myriads of questions about European foul brood. So today I'm going to go through a little bit of the history of research and why we have so many questions still, kind of the stumbling blocks scientists have come across, and then kind of transition into more modern research, what we know now, and kind of where the current research is headed. And then we'll end with some management. Dr. Meghan Milbrath will be crucial in that part. So what is European foul brood? So this is a bacterial disease affecting honeybee larvae. So this is the bacteria, Mosacoccus plutonius. The bacteria makes its way into the larva in the first few days of life. It replicates within the larva's midgut. The larva changes from a pearly white, which is a nice healthy larva. It starts to get kind of off color with early symptoms. Eventually, the larva twists within its cell and then devolves into kind of this various stages of goo after it's dead. So as a veterinarian, when I come across a disease, I have a few basic questions. First, I want to know, how prevalent is it? Is this something that I'm going to be dealing with consistently? Is there regions of high prevalence that are higher risk that I need to know about? How is the disease transmitted? Is it on my hive tool? Is it just by the bees going from hive to hive? Is the pathogen able to survive on my bee suit? How careful do I need to be? When a hive is diseased, what's the prognosis? Is the hive always going to tank? And with the case of European foul brood, we'll learn there's various prognoses based on a number of factors. How is it treated? With European foul brood, we have management options, but the treatment option that we have is oxytetracycline. We'll talk a little bit about that. And then I want to know what kills it. So most of these questions have very few answers despite 130 plus years of research. So we'll go into a little bit of the history of why that is. So the first description of foul brood, so foul brood more generally, happened many, many centuries ago, but the first formal description was in 1885. So they talk a little bit about previous, beekeepers have been dealing with this thing, foul brood. The larvae are dying. They don't know why. It stinks. So Cheshire and Shane were two scientists working on this disease in the UK, and they published a paper in 1885 claiming to it was the first formal description that we really have of foul brood. So it was an attempt to kind of determine what the cause was and to kind of formally describe all the symptoms you would come across. This was before there was a delineation. Now we know of European foul brood and American foul brood as two specific diseases, but this was kind of before there was that delineation. So they claimed to have found the cause of this disease, and that it was a spore-forming bacteria that they found, and they called it Bacillus alveoli. So there was a caveat in their paper, though. The organism, when they fed it to healthy colonies, did not reliably cause disease. So when they were doing this research, this was at the very early days of infectious disease research. 5 years later, this guy, Robert Koch, would publish what's now kind of known as the fundamental basis for how you determine the cause of a disease. And he published these four postulates. So the first one is if you think there's a cause of a disease, a pathogen that you're interested in, you have to find the microorganism in abundance in all animals that have that disease and not in healthy animals. Pretty simple, right? The microorganism must be isolated from a diseased organism and grown in pure culture. So this is a way to make sure we only have one bacteria and not many, many bacteria. So to do this, we take the pathogen from the diseased animal, we spread it out on a plate in the lab, and then we take one colony that should be a monoclonal, just one kind of dot on the plate, and then we grow that up in culture and then assume that that's a pure bacteria, just one strain. So we grow that up in the lab, we feed that to a healthy organism, and it causes the same disease. That's the third. And then the fourth is we can retrieve an identical organism from that diseased animal. So with all four of those, you kind of have a pretty good confidence that you've determined the cause of the disease. So they did not find this organism in abundance in all of the diseased larvae. They were able to grow something we now know as Bacillus alveoli. It did not reliably cause disease, and hence they were not able to consistently recover the bacteria from diseased animals. So many of these postulates still remain, right? So kind of looking back on this, and it's pretty clear from the photograph, all the larvae, this is pretty consistent with what you'd see with European foul brood. But the difference between these two is all the capped brood. So all the stuff that's got pupa under it that's capped here, a lot of it's kind of damaged in the left illustration. And on the right, it's pretty pristine, right? So not a lot of the older brood is affected. It's pretty clear that they're conflating both brood diseases when they described this, but when they went to study it, they were only looking at one. So it wouldn't be for another 10 years that a American scientist working for the USDA by the name of Gershom Franklin White started studying a disease that was severely affecting honeybee colonies. So this was killing colonies outright, and it seemed highly infectious. When he studied this, he noted that the symptoms were very different from a lot of the symptoms that they were describing, especially that photograph that you see in that Cheshire and Shane article, right? With this disease, G.F. White was able to pretty consistently recover a spore-forming bacteria from all the diseased larvae and fulfill Koch's postulates by feeding that back to larvae and causing this disease almost all the time. He would go on to kind of formally describe this disease because it was first formally described in the United States. He called it American foul brood, and he called the bacteria that caused it Bacillus X, which eventually renamed Bacillus larva. And on the bottom, you have kind of his drawings of the different stages of this bacteria as it goes through and eventually ends up as a spore forming spore stage at the end, which is in the infectious stage. Koch's postulates were fulfilled for American foul brood relatively easily. So what were they describing exactly in Europe that was affecting these younger brood, right? And maybe there's two diseases. They did come across a lot of this disease that was affecting younger brood in the United States too. So the same scientist, G.F. White, continued on to characterize this disease. What he did that's different, however, is he noticed that larvae were diseased but still alive. So like this image you see on the top with the discolored larva, that when he looked inside the midguts of those, in all cases, he found highly abundant bacteria. And this is kind of a drawing of the bacteria that he found. And he determined this to be the cause because it was so persistently evident. But when he sampled these decaying, kind of melted larvae, he noticed a lot of other bacteria were present. A lot of like just environmental bacteria were taking over, colonizing, and it was difficult to isolate any one specific thing. So this was 1912 when he formally described this. But the problem was, try as he might, he could not actually get this organism to grow. He could grind up larvae, feed it to other larvae, and cause the disease pretty predictably. But when he would try to grow it on all the cultures he tried in the lab, nothing would grow. So this was his apiary in New York where he was doing his work. So we found the organism abundant in all diseased larvae, couldn't grow. So scientists spent the next 4 decades trying to grow this bacteria in the lab. It wasn't until 1956, again, a UK scientist, Bailey, was able to come up with a very specific concoction that has potassium supplemented and all these different requirements. And in the lack of oxygen, he was able to get this bacteria to grow. And this is one of the first photographs we got of this bacteria. So it forms these kind of unique long chains. But another another hurdle. So he could grow it in the lab, but a lot of times when he fed it to larvae, it failed to cause disease. So the virulence was lost, right? So back to Koch's postulates. We've got the organism in abundance. We can grow it and isolate it in the lab, but it's not consistently causing disease. So more questions, right? It wasn't until really the 80s that scientists were like pretty certain that this was the cause, even though they couldn't get the pure culture to cause disease. The next real breakthrough didn't come until 2012. And before I talk about the next kind of breakthrough, I want to do a brief overview of bacterial genetics. Here's a bacteria. Within bacteria, we have a piece of DNA, usually it's a circle, called the chromosome. And this has the instructions for all the things the bacteria needs to do to survive. Like all other life forms, bacteria wants to eat and reproduce, right? So it eats, it reproduces. When it reproduces, it basically clones itself. So it makes a bunch of copies. One of the problems with this, if you're looking to adapt to new environments, is you have exact copies. If you encounter things you're not ready for, your population can collapse. So to deal with this, bacteria have a strategy for creating diversity. So sometimes they make mistakes as they're replicating, and sometimes, not always, usually these mistakes are deleterious. So they cause harm to the bacteria and the bacteria dies. But on occasion, they can be in a gene that changes how it functions and gives the bacteria an advantage. And then in those situations, the bacteria can adapt. So bacteria have also another way of increasing their kind of genetic diversity to allow them to deal with different challenges. And that's through what's called horizontal gene transfer. So outside this chromosome, which is their main set of instructions for survival, they can also gather DNA from the environment or transfer DNA from one bacteria, even between different bacteria species. So here we've got these kind of extra pieces of DNA. These ones are called plasmids. It can transfer to another species. If there's genes on that chunk of DNA that the bacteria finds useful for certain situation, then the bacteria will keep those. So a lot of these are a lot of the bacterial changes are driven by all the pressures that the bacteria is put in different circumstances, right? So if you think about all the circumstances that a bacteria has to go through to cause European foul brood. So we've got bacteria has to make its way into a larval midgut, relatively oxygen-free environment, kills the larva, but then it needs to transfer itself from larva to larva, right? So it needs to survive also within the mouth parts of adult nurse bees as they clean out that larva and then they go feed other larvae. So it has to survive also in that nurse bee, but it also wants to make its way between hives, right? So it's got to get transferred to a foraging bee. The foraging bee's flying. It's a more oxygen-rich environment. More selective pressures are being imparted. Then it has to make its way into a neighboring hive, a nurse bee in that hive, and then it has to survive the incredibly antimicrobial environment of royal jelly, which gets fed to a healthy larva. So at each of these stages, the bacteria is adapting, creating small mutations, and it can allow it to create sort of a fingerprint. And eventually it ends up happy. So this is a cross-section of a larva that's infected with European foul brood. As you can see, the midguts, all this purple stuff here is Mosacoccus plutonius. So it's just packed with this bacteria. The bacteria is very, very happy within larval midguts. So how does this help us understand EFB? So this can serve as sort of a fingerprint. So as it's going through all these changes, the bacteria is forming a unique genetic code, right? And this can allow us to track spread. So obviously we were doing this with COVID quite a bit. But DNA also has all the blueprints for the bacteria to survive, right? So what does it need to grow? How does it survive antibiotic treatment? How does it even kill the larva? So this is something that's still the subject of study. We don't entirely understand how this bacteria is killing the larva. And then finally, what pathways can we use within this to kill the bacteria? So this is an example of how the kind of fingerprint can help us understand spread. So if we got 5 beekeepers and they all have European foul brood outbreaks, we can go into those beekeeping operations, recover the bacteria, and then we sequence the genetic code. So with all those sequences, we can come up with a phylogenetic tree that tells us how all those different strains from those different beekeepers are related. So you can see here this kind of tree on the lower right. We got the red and the beige. Those are closely related. The blue and the purple, those are closely related. So from that, we can kind of infer there was some common source between beekeeper A and D and some common source between beekeeper C and E. So somewhere down the line, those two, whether it's exchanging bee packages, equipment, somewhere there was a common shared source of infection. Beekeeper B, on the other hand, this green one seems to be kind of removed, so maybe another source was involved with that. So this was actually used in Europe. So in Europe, they have a fairly low abundance of this disease. The prevalence, I think, is around 1%. So this was a study that took place over many years, looking at the genetic fingerprints of European foul brood in outbreaks. So on the left, we have kind of globally, each of these circles is kind of a different unique variant of this bacteria. And then on the right, we have kind of these groupings. So you can see each different color and shape is a different strain. So that corresponds also to strains that are, not the same color on the left. But so when we're looking at the map on the right, we can see like the green ones, like there's clusters in the south of green. If you go up farther to the west and the Midwest, it looks like you have clusters of red. On the east side of the area, we have clusters of kind of the light blue. And then you can see kind of the green goes up towards the red, but not necessarily over towards the blue, and that all of them kind of merge and mix up in the northern part. So with this, we can kind of see where strains are coming from and then kind of where they're merging. And this allows us to kind of better understand how beekeepers are involved with transporting these strains across these large spaces. What else can DNA tell us? Like I mentioned, this has got the blueprint for a lot of the characteristics of the bacteria, right? So, like I mentioned, the next breakthrough came in 2012, and this was when in Japan they noticed an unusual variant of this bacteria. And this was before they knew anything about the genetic sequences. They saw that this bacteria could grow in the presence of oxygen, which was very unusual for Melissococcus plutonius. So they looked at the genetic sequence and saw indeed there were actually two unique genetic groupings of Melissococcus plutonius. In this illustration here, if you look on the lower left, you have this red circle. So that's the one that they could grow in oxygen. They termed that atypical. So that genetic grouping seemed to grow faster, could grow in the presence of oxygen, and was pretty far removed from a lot of the other previous strains that they'd encountered. So those ended up being grouped in the typical. So the next question was, all right, there's like a lot more genetic variation in this bacteria than we had assumed. They wanted to know, how does this impact the larva? So what they did was they fed strain from this grouping, this typical grouping at the top to larva, and a strain from this atypical grouping to larva. And they noticed that in the case of the typical, sometimes it didn't cause any disease at all. So this is what's called a survival curve. It's got basically the percentage of larvae that survived this infection at each day. And you can see the control is the gray line here. You see almost all the larvae survived when they're growing them in these petri dishes. The one that says DAT 606, that's the typical strain, more survived and that had the bacteria. So it had no impact. But when they fed them the atypical strain, they were able to predictably cause disease. And so this was kind of the first fulfilling of Koch's postulate with this atypical strain. But why was this typical strain not causing disease? When they looked closer at the genetic code, they noticed that there's these plasmids. And something that commonly happens when you're growing things in the lab, after you repeatedly grow them over and over and over again, if the bacteria doesn't have any reason to keep those plasmids, sometimes it won't replicate them. So they noticed some of the typical strains would cause disease that had these plasmids. The atypical strains also caused disease and had these plasmids. When they grew them over and over in the lab, they would lose these plasmids, these extra chunks of DNA outside the chromosome. They noticed when the typical strains lost the plasmid, it lost its ability to cause disease. So this may be why back when Bailey first started growing this, he was likely dealing with these typical strains, and through the growth of them was losing these plasmids, and it was losing its ability to cause disease. However, the atypical strain causes disease anyway. So there's something in the actual genetic code that's able to cause disease, whereas the typical ones seem to rely on this extra DNA. So a complicated picture, right? So we kind of know two big kind of genetic clusters of bacteria that causes EFB. There's this typical one and this atypical one. The atypical one seems to cause disease all the time. Typical one, sometimes it does, sometimes it doesn't, right? So what about the United States? A lot of this research so far has happened in Europe and other countries. So we undertook a 2-year surveillance study back in 2021. It went from 2021 to 2022, and we enrolled tons of different colonies from tons of different beekeepers of all sorts. We found that we could find the DNA of Melissococcus plutonius in about 30% of the colonies. The complicated thing here is that in most of the ones that we found this in, we were finding both the typical and the atypical present within the same infected honeybee colony. So this was very common. This was also observed in Japan and Mexico. So super abundant disease, way higher prevalence than they were seeing in Britain. And there seems to be multiple strains often within the same honeybee colony. We continued on to look at kind of the seasonality of this. We wanted to know, was there times when this peaks? The seasonality of this disease has been described for quite a while, but it's never been kind of formally recorded until recently. So this is basically the percentage of colonies we tested within each month that had the bacteria present. So we could find the DNA of the bacteria within the honeybee colony. So you can see in June, over 60% of the honeybee colonies that we tested had this bacteria in it. So when we're to think about clinical disease, though, it's a little bit lower, but not a ton lower. In June, we still had over 40% of honeybee colonies had clinical European foul brood. So this is impacting the colonies in a very real way. And something important to notice about this also is that this gap between the red and blue, so these are colonies that had the bacteria present but weren't showing any symptoms. So these were kind of asymptomatic carriers of this pathogen. So we have really high abundance of pathogen infection, and we have really high abundance of the actual clinical disease. So what does this tell us? So in the United Kingdom, it had relatively low abundance, and they could see, okay, we have an outbreak, we have a strain of bacteria, and we can see its relationship within the landscape. But within the United States, we have huge abundance, and what's more, we have multiple strains within each hive, so it looks more like this. So it becomes pretty difficult to determine what the direction of spread is when it seems like everything is everywhere, right? So this is kind of the picture of a disease that's gone largely out of control. So treatment. Is treatment still effective? So we've been using oxytetracycline to treat this disease since the 50s. And like we know with basically all other diseases that we've used antibiotics consistently with, it's only a matter of time before the bacteria evolves these mutations that help it survive, right? There has been some recent reports from Canada and Japan that show some mild resistance in the lab to this antibiotic. One important caveat here is the resistance in the lab might not mean anything. So I want to go through really quickly how exactly we test resistance for this antibiotic. So this is what's called a microbroth assay. Within each of these wells here, we have the broth that the bacteria likes to grow, right? As you go up a column, you have increasing concentrations of antibiotics. So you can see the 128 is the highest concentration on this top row, and then on the bottom row, we have the broth with no antibiotics. What we basically do is we put the bacteria that we're interested in. So we have a bacteria strain that we got from an infected colony. We put it in each of these wells with each of these concentrations of antibiotics, and then we just see at which concentration does it stop growing. So this was Mollisococcus plutonius. This was pretty consistent for most of the strains that we tested. And then this is our positive control. So this is a bacteria that we know has evolved mechanisms to deal with this antibiotic. So if I was going to read the results of this, I would say, okay, at the very lowest concentration of antibiotic, that's inhibiting the growth of this bacteria. For our control, if I was going to read the control, the minimum inhibitory concentration, they call it, the MIC, is 64. So this would be considered highly resistant to oxytetracycline. So that's generally how we test for this. But there's a caveat with this test, and this was something they noticed in the study in Canada as well, that within these higher wells where we're not seeing growth, say up here at 32, if we pull material out of here and we grow it, we can reliably grow the bacteria. So the bacteria is not growing within these environments, it's inhibiting the growth, but the bacteria is not dying either. So it's persisting. So this may go a long ways to explain why we've been treating this disease with this antibiotic for so long, but it still seems to be a really, really high prevalence, right? So that led us to kind of this next portion. So this was a project taken on by one of our summer research students, who's now a veterinarian as well, Robin. And so we think that this bacteria is probably persisting. You know, we think about this a lot with American foul brood, these spores, they last forever on the equipment. You don't want to get stuff used because it might have stuff. You don't want to bring any infection into your operation. But nobody really talks about this with European foul brood, right? So we took two different strains, one of the atypical and one of the typical, and then grew it up in the lab, and then put the bacteria just on these different surfaces to just see, you know, within the honeybee colony and, you know, on metal. So we used steel washers to replicate a hive tool, or on cloth to replicate a jacket. So this is you can see on the bottom is these all the different materials that we put the bacteria on, and then waited. What we found was on all the material, these strains survived for at least 2 weeks. The typical strains seem to have less viability, less of them survived, but they all survived for the 2 week study. She was also nice enough to make us a batch that we could wait 1 year and test, how does it last between seasons, long term. And what we found was the atypical remains viable on all surfaces a year after study. And this is a video I took of one of these strains of atypical Mosacoccus plutonius growing in culture, and it seems to form this kind of matrix that we're not entirely sure what it is, but it may have something to do with its persisting. So this bacteria hangs out on material for a very long time. So where are we now? So we have these main questions that I kind of threw out at the beginning. How prevalent is it? It is highly prevalent, both bacteria, infection, and disease, at least in Michigan. We'll say at least in Michigan. How is it transmitted? Still a topic of study, but we do know that it hangs out on the material that we tested for a very long time, at least a year, right? So it's very likely that there's mechanical transmission between hives. How much of it is surviving on these surfaces is something we're still looking at. So maybe it's not enough to cause infection. What's the prognosis? I didn't mention this, but we did a study following colonies with European Foulbrood. And while there's a lot of different outcomes that hives can have based on kind of different strains, we found that overall colonies that were severely affected with this disease had a 54% reduction in colony growth. So it definitely impacted the colony pretty heavily. Treatment, like I mentioned, oxytetracycline seems to still be at least as effective as it was when they first started treating, but this bacteria may be outlasting the antibiotic on our material. And then the last question, if it's hanging out on our material, what can we use to kill it? And so this is something we're currently working on. We're testing a number of different things, sort of thinking about widely accessible things that people can use to decontaminate their equipment and help reduce the pathogen within their apiaries. So with that, I will turn it over to Dr. Milbrath. Sorry for all the bad news. Hopefully we can have some better news on how we can effectively manage this disease, and looking forward to questions. So
Yeah, thank you, Peter. And I am not going to make it get any better. So we should have warned you all that this is a night of depression. Hard to grow, hard to research, hard to manage in the colonies. So that was a really lovely background of where we have been in trying to study the pathogen. And then now I'm going to go into the more hands-on side of what you actually do if you have it in your colonies. One thing I did want to point out from the prevalence study is that when we were looking through these almost 1,000 colonies for the signs of disease, we were in very large migratory operations. We were in sideliner operations. We had hobby beekeepers send in samples. I've seen it in operations where there's 2 hives, and I've seen it in operations where there's over 10,000 colonies. So it is really widespread, but it's widespread across the board in our state, and we have samples from other states, and we hear it's definitely not uniform, but it is quite serious. So there's a pretty good chance that you could come across it in your operation. So what should you do if you suspect you have EFB in your hive? So the first thing that people do, at least the ones who call me, is panic and want to burn it all and just quit beekeeping. And that is generally not our recommendation. One of the things that I do like to recommend is, you have to figure out which disease you have. So whether or not it is actually one of the Foulbrood diseases, and then you deal with it, and then you just get back to beekeeping. But another nice thing that you can do is actually take the time to show others. One of the things that we're going to talk about is in diagnoses, a lot of it is visual identification. And Peter and I and Heather, our technician, and the people who work with me, we've probably seen, thousands and thousands of cases of EFB. I feel completely confident that I can diagnose EFB visually. And that's a skill that it's really useful for other people to do, because that way you can get your answer. You look at it or you look at a photo, and then you could know what action to take. And we want veterinarians to be trained as well. So Obviously, we want to be safe in this context. So you can see we have students coming out with nitrile gloves on, and the person who owns the hive is actually handling the hive, but allowing people to see it with their eyes right away is an incredibly useful thing that you can do. So you do want to confirm that you had it. So the main one, like I mentioned, is the visual signs. So Peter talked about these with the tracheal signs. A lot of it, the official term is like loss of internal pressure, but basically we call it melted. And when we talk about the trachea, it almost looks like you can see the skeleton. So in that photo on the left, you can see kind of the actual trachea in, it looks like the bee starts to get a little translucent. That twisted corkscrew is very characteristic of European foulbrood. The melted one you can see with other things, especially parasitic mite syndrome. And then this one, you can see the trachea really obvious on that scale on the bottom. One thing that you note with the photo is that it's very, very diverse. So you can have yellow and brown and green and twisted and tracheal and all of those things on the same frame. And you'll have bees that are starting disease and ending disease. Whereas with things like American foulbrood, it's much more diverse. And even with American foulbrood, it's much more consistent. With parasitic mite syndrome, it's much more consistent. With sackbrood, it's much more consistent. EFB, the name of the game, is just very, very diverse. And so here's another one. You can see some chewed down pupa. We actually have a co-infection with chalk brood on this frame. And then this one, you can actually see the holes in the cappings and starting to dry down into a scale. And then at the very bottom, right below that, there's two cells. It is kind of hard to see, especially if you have a small screen, where there's
Oh,
actually a rubbery scale.
yeah.
And so a lot of times people will think that is American fall brood, and you can get a co-infection with American fall brood. We've seen a lot of co-infection with sac brood as well. But with European fall brood, those melty larvae will kind of dry down, but you can actually pull out the scale. With American fall brood, it'll dry down and it'll be kind of painted on. But again, you can see on this slide lots of different things going on. So again, lots of different colors and lots of different signs all at the same time. So as I mentioned, if you suspect that you have it, especially if you're in Michigan, one of your options is to actually use our questions. So if you go to, this is our pollinators.msu.edu, you hit on the questions tab, it will take you to this form. And one notice of this form is that you can upload photos as well. So if you suspect that you have European foulbrood, you can take some photos. If you just don't even know what you're seeing, you can take some photos and upload them and can look at them. And then we also have this website that I'm incredibly proud of, which is the diagnostic tools for honeybee diseases. And this is something that we made this last year. And the target audience was veterinarians, but it's also for beekeepers as well. And there's lots of photos where you can identify what European foulbrood looks like, what other diseases look like visually. We have all of the different diagnostic tools, so ways to use them, how to select the correct samples for each one, common pitfalls that people do, and little tips and tricks for diagnostics. It even has very fun games on there. So if you want to be entertained while you learn how to select the right age larva, this is the site for you. All right, so one of the diagnostics options is the lateral flow devices. This is a little difficult to talk about, so we're recording this in 2024. You can sometimes still find them available. The company who produces them is located in the UK. There are some issues importing them and having it economic for them to import them and meet all of the USDA requirements. But there are some places that can, you can bring them in under a research permit and distribute them as part of your research or use them for part of your research. They're just not currently available for sale at the moment, but that might change. But basically, this works just like the COVID test. You take a small bit of larva, you macerate it in the buffer, you use a droplet, put it on the test device, and you see a common positive result here where it's pretty faint, but the T is the test line and the C is the control line. So this would be a positive. Peter and I actually did a study with, I think, 90 or some samples where we tested it on because this hadn't been validated in the US. And so we used samples of different strains from around the US and found that it did work well for us. One thing that we were very, very careful about when we were using this device was to select larvae that were very, very recently infected. So I showed you all those pictures of those gross, like dried down, blackened, and yellowy ones. And again, you can learn this with a fun game. So here you can see some that are just starting to be corkscrewed, but still look like they're still alive. And those are the ones that you generally want to select for diagnostics, be it using this lateral flow device, sending to Beltsville, doing microscopy, whatever you're doing, it's these earlier ones or earlier stages of infection. And the reason behind that is we actually confirm the disease by looking for Melissococcus plutoneus, and that bacteria gets taken over by a bunch of secondary bacteria later in infection. So when you first have the disease, the bacteria is really, really replicating quickly. And then as the larva dies, the bacteria can't continue to replicate without the food source. And secondary invaders come in, or ones that will eat the dead bacteria or the dead larva, and then you don't really see Melissococcus anymore. And so you can get a false negative in that case because you're just kind of too late in the infection to find it. So one really key message that we've been working on is making sure that when people are sending things in for diagnostics, that they are sending in something that's kind of in the earlier stage or a wide diversity of different sample types. The other thing is you do want to rule out Varroa. So you would do an alcohol wash at this time. We have definitely seen colonies, we talk about that seasonality. We've definitely seen colonies that have both parasitic mite syndrome and they go right from EFB into parasitic mite syndrome. But we do get a lot of calls about people suspecting they have Foulbrood, in September in Michigan. And in most, even though we can and we have seen it, in most cases, that's going to be parasitic mite syndrome caused by Varroa-associated viruses. And then, as Peter mentioned, it does have a very strong seasonality. So where we are in Michigan, it is really late spring, early summer. If you see it in the late summer, fall, then it's going to be the parasitic mite syndrome. In Michigan or in these northern states, if you've got really diverse looking larvae in the midsummer, it's a really strong chance that it's European foulbrood because also, remember, Peter talked about like it's in 1 out of 3 colonies. And so that's the likely thing as well. And I mentioned we see thousands of cases of EFB every year, and I saw 1 case of American foulbrood this year. And that's, 0 to 5 cases of AFB every year to thousands of EFB. All right, and then you can always confirm by sending a sample to Beltsville. This is really useful. So Beltsville is where our USDA laboratory is for beekeepers. It is a free service for beekeepers in the United States. I didn't put any instructions on here because every time I want to give them out, I just Google send bee sample to Beltsville. And it'll take you to a page on how you send in a sample. It is really, I found an article from the 1930s that used the exact same language, so they haven't updated their website since the 30s. You basically want to send a pretty large amount. And the reason it's outdated is a little frustrating for beekeepers who use plastic foundation. It talks about using a huge chunk, and we tried to figure out a way to take a huge chunk in off the plastic. It's really hard to do it without mashing it. If you have beeswax foundation or your foundation list, you can send a nice chunk in, and that allows Sam, who receives the samples, to actually use visual identification himself. And then they'll actually use other tools to confirm in the lab. So again, if you can send a large chunk of comb, that's ideal. Otherwise, you can use a swab. But when you're doing that, you want to aim for those larvae that are just showing the early signs of disease. All right, so that's how we diagnose. Again, it's a lot of visual identification, a lateral flow device if you can find it. We also have guidelines for microscopy if you really want to get into it. That's on our website if you wanted to do that. yourself and then sending it for confirmation at Beltsville. But the visual identification is considered sufficient in most cases. So one of the things that Peter mentioned is that there's a lot of inapparent infection. So that means we can find the pathogen in a lot of colonies that look perfectly healthy. And so what we want to think about is not treating this disease as a hive level, but as kind of a yard or really an operation wide issue. So there's a lot of data from the early 2000s that talk about when you have one symptomatic colony in a yard, they're going to be able to find the bacteria, especially in nurse bees, and sometimes even in larvae in colonies that look perfectly healthy. So this is really important when you think about treatment. One of the constant recommendations that I see from people is, oh, take that hive and put it into a quarantine yard. So if I had 10 hives in my yard and one of them has signs of disease and I button it up, put it in my car, do all this work and take it to a quarantine yard, I likely have not done anything to protect the bees in the rest of the yard. So that's a lot of effort for no effect. Or if I'm really, really concerned about just treating this one with disease and I just get antibiotics for this one and I'm really worried about just this one, I want to be aware that, the other ones it'll kind of feel like it moves through the yard, but in most cases, the pathogen is likely in the system already. And a big reasoning for that is because it is in the nurse bees so much and it is in the adult bees. So when we have drifting and we have robbing, and as Peter mentioned, it's so persistent on equipment when we're making splits, it's very, very quickly transmitted within the yard. And again, that's in really high contrast to American Foulbrood, which doesn't get into the nurse bees quite as much, where we can see, one or two hives in the yard, but you don't expect to find it throughout the entire yard. All right. So it's really hard to discuss about what you can do. We don't have set guidelines for treatment of European foulbrood. It's not a reportable disease in the United States, though it is in some countries in Europe and elsewhere. So we do have some options, and really what we recommend for options very much dependent on what you can logistically do well, and also what are your chances for reinfection. So, one option is you just burden your operation and you quit. That is an option that does reduce your amount of disease. And I have talked to beekeepers who do want to burn everything and start over. Well, that's not going to work well if you're in a city where there's a huge hobby beekeeping group, or you're next to a really, really high density bee area where there's just lots of people purchasing bees and moving bees. Your chance of reinfection is really high. So you'll spend a lot of money and not be able to eradicate it. If you've got, you know, hardly anybody else in your area, you're very far removed, you've only got a few hives, and you just don't want to deal with it forever, then you could try to do eradication. And it might be a combination of euthanasia and destruction. That has been the method that is used in some countries in Europe. And in those cases, they are able to eradicate it for a time period. There's no case that I know of where a region's been able to get rid of it permanently. So it's something where you could spend a lot of money and then not see it again, but you could spend a lot of money and effort and be really dealing with it a lot. So Peter mentioned what we know about the antibiotics. I'm going to talk specifically about antibiotic control, but that is one of our other options. You can do a shook swarm, which I'm going to explain. You can do both. Or I have the term watchful waiting. So one of the things about European fall brood is that it does, because it's seasonal, we do see the signs go away in a lot of cases. But what we want to think about is if we choose not to do anything in this colony, kind of the ethics around the situation. So, and what we're considered about ethics is the life and welfare of that colony itself and the risk of transmission to other colonies. So if we don't want to have a colony that's going to get worse and worse and worse, have more and more dead and dying larvae, have a higher pathogen risk in the yard and have worse health outcomes. That's not something that's acceptable in other animals. It's not acceptable in bees. But we also don't want to burn a lot of equipment, and we also don't want to use antibiotics unnecessarily. So a lot of it is I'm going to talk about how these treatment options work kind of, or how you can use them with different size operations. All right, so one is the shook swarm method. This is a method that there is actually data to support. The idea behind it is you take the bees and you shake them onto brand new equipment. And so you're going to be using, and when I say new clean equipment, you can see that this is straight foundation. So the idea behind it is you've got all these nurse bees or all the bees, the adult bees that are in the colony, shake the bees onto foundation, and then whatever the bees have in their crop, they can't just spit it up into drawn comb. And so if they have pathogens in the honey that they've digested or that they've ingested, they actually have to fully digest it and either turn it into wax or defecate outside the hive. But the idea is that they have to actually draw out the wax. The other thing is you get a nice long brood break because the bees take a while to draw the wax for the queen to start laying. That's called a full shook swarm. There's also a version with a partial shook swarm where you just take out the frames from inside the brood nest and kind of give them a brood break, but you don't worry about removing everything from the hive. So the first step is to set up the new equipment. And then the second step is to shake the bees. So this is actually, we were doing this with a hive with American Foulbrood. You can do it with AFB as well, as long as your state or province allows it. But you just shake the bees and then you take that equipment and you put it in a place where it's not going to get robbed out. So here we just moved all the frames, covered them on both sides, and then we transported them. In most cases, you're going to be destroying the frames. You do have the option to treat all the colonies in the yard with the antibiotics. So the idea with that is that if you had bees that have the bacteria on their mouth parts, once they start laying larvae again that are in that susceptible range, those larvae aren't going to be infected right away. So you're kind of protecting that first generation. And then by the time you have the antibiotics have worn off or you're done with your treatments, which is about 15, 20 days, then those bees that still have it are going to be gone. You also want to think about if you know that it's not widespread through your operation or you do have different yards, you can think about using that as a little control yard. So this was actually a case with AFB, and the two smaller colonies were a split that had AFB, and the third hive that was in there obviously didn't. It had all the honey, and I just distributed the honey amongst those other ones and kind of just watched it for a year. So the nice thing about the shook swarm is that you're getting rid of a lot of the pathogen, so your chance for reinfection is less likely. You can decide what to do with your old equipment. If you are using regular foundation, you can burn it. If you've got plastic foundation, here you can see we've got a contractor bag, and we're taking these frames to an incinerator, and you're actually properly disposing them at a landfill if you're not in a place where you can burn it. There are a couple other options as well. So as Peter mentioned, we're working on disinfection. We don't have good recommendations. So a lot of times people say, well, can I dip it in bleach? And the correct answer is I don't know. There is sterilization that, especially even for AFB, there's been a lot of work on using heat plus time, or there are some facilities that do gamma radiation and you can actively sterilize the equipment. It is not cheap. And one of the things about that is, it is high labor. You could clean out that equipment. You want to make sure that you're weighing the cost with your chance of reinfection. So it's not going to, the second you put in new adult bees, there is a chance that, they're going to come in with all of their pathogens. Another thing that we've been working with at MSU this year with another veterinary student is setting up a boundary system. And I'll talk about this more on the next slide, but what we've done is kind of cleaned out some of the yards that have had a history of EFB But instead of burning all that equipment or doing shook swarms, killing everything that even the stuff that looks good, is we set up, we make a clean yard where we shook swarm everything, and then we put all of the stuff that's not bad into a yard that already had EFB. And so we're kind of systematically moving through and cleaning out particular yards. So you don't necessarily have to destroy everything in the case of EFB like you would want to do if you had American Foulbrood. All right. And I'm not going to go through the details on this, but if the colony is really small and it's really late in the season, then you wouldn't want to do a shook swarm. And in that case, you would want to euthanize the colony. There are going to be guidelines coming out soon through the American Veterinary Medical Association. And the ones that are recommended are usually using an insecticide or using diesel fuel. And there are a lot of resources. Those are commonly used by governments. I would say the diesel fuel is in cases that they've had to do a lot of depopulation. So there are guidelines that are accessible for that. So the shook swarm method, it does work, but it's not foolproof. So I've used it quite a bit, and you can see it come back the next spring. With the partial shook swarm, I've had it come back even the same year. So it is really a balance between effort, loss of equipment, and you're reducing the pathogen risk. I do have to say, though, if you're opening a hive and it is just disgusting and it looks like everything is infected in there and those larvae are gross, it's kind of hard to leave it in there. There's just a lot of pathogen in there. And so that would be a case where it just does kind of feel right to remove that. And so that's not, it's never really a bad option. You do take a hit. So in terms of honey production and queen loss, so this year at MSU, we looked at yards that we shook swarm just the colonies that were sick compared to yards where we shook swarm the whole yard. We don't have data because we want to see what happens in the spring. So stay tuned for next year's EFB webinar. But what I can say is that it felt terrible in the yards where we did the whole yard. We were a little late in the season. It felt terrible to do these shook swarms on perfectly healthy looking colonies, and there's just like nectar dripping everywhere. They looked great, they were in production, but for the ones that are really sick, it feels very intuitive. For the ones that look healthy, it doesn't feel as intuitive. It's hard to know how much pathogen is in kind of that yard and all that equipment, though. You really can't tell necessarily by sight. So it isn't maybe a good system for all the time. So here's my personal opinion on when you would kind of use it, but I just wanted to make sure that it was clear that there isn't like a standard set of regulations or recommendations. So one thing is it does have to be early enough in the season so your colony has time to rebuild. They have to actually draw out the wax. You'll have to feed them as well. And this is something, though, if you think about if you want to be someone who is selling nukes or selling queens or having equipment leave your operation, then being really, really systematic of removing frames that have had signs of disease, I think is really important. Like I said, it's difficult to do the whole yard, but if you can do it at a yard level, that's ideal. And so what it looks like at the yard level or at the larger scale is one, what I mentioned is you can set up a boundary system. So you've got a clean yard and a dirty yard, and your equipment only flows in one direction. So what we did is we picked a yard that had 2 yards that had EFB, and one of them got all brand new equipment. We shook all the bees onto new equipment, and then we got rid of all the comb that was kind of gross or had lots of pollen in it. And then anything that had like healthy looking brood or good honey went to that yard that we already knew had melissococcus in it, or we already saw signs of EFB. So that way we had a yard that we knew had really low pathogen risk, had a pretty low chance of it coming back next year. And the other yard wasn't really worse for where we weren't really making it worse. We were just kind of keeping this boundary system, which again, with EFB, because it's so prevalent and persistent, we're not really in a place where we can talk about eradication yet. We are really talking about control. So if you know you have this pathogen, how do we minimize it and we minimize the spread and we minimize the damage to our bees? Another thing is being really deliberate with comb replacement. So we work with a beekeeper who every year shakes a bunch of packages off of their own bees and then scoops those packages into brand new equipment, adds a new queen, and then has kind of a set of old equipment and a set of new equipment. And they use those new hives for honey production, and that old hives go out to pollination. And so instead of when you have to replace frames every year, instead of maybe giving everybody one or two frames, trying to think about if you can set up a clean yard. So if you're replacing 100 frames every year, it might as well be a nice new yard where you can kind of keep the pathogen down versus just one frame per every hive. All right, so that's the shook swarm. Again, it can be used with or without antibiotics. Your other option is antibiotics. And the system, if you wanted to use antibiotics, is to talk to your veterinarian. We have a training program in Michigan, and it's in a couple other states where we are training veterinarians. The Honeybee Veterinary Consortium does have a listing for people who are veterinarians in the state. We are going to have a listing shortly with our Department of Ag. You can, if you find a vet and they say, no, I don't work with honeybees, you can give them our contact information in Michigan, and we will help them so that they can get to a place where they can prescribe antibiotics without losing their license. That is their major concern, is that they have to meet certain criteria in order to work with an animal so that their license is intact. And so we've got a lot of resources for them and are happy to work with vets. If you're using antibiotics, Peter mentioned oxytetracycline, or also known as terramycin, or that's one that's sold as TerraPro or TM50. That one is the one that is labeled for control of EFB. So it's not labeled for treatment, it is labeled for control. And that is what I said, is like we're not necessarily going to eradicate. It doesn't kill the bacteria, it just prevents it from replicating. There are other ones that have been shown to work in the lab. And so hopefully there are more treatments in the pipeline. You do have to get an order from a veterinarian to access them. So I'm using the term order. That's a general term that refers to either a veterinary feed directive or a prescription. So it's something that the veterinarian has to write, and then they submit it, and that allows you to have access. The only different from your side is where you get the antibiotics from. So if it's a veterinary feed directive, then the vet's going to email that to one of the supply companies that's also a licensed feed mill. And once that's on file, then you're able to order the antibiotic from them. If it's a prescription, then you get it from a pharmacy. So the hard part, though, and the reason that there's a little star there, is because it is sometimes hard for pharmacies to have the correct amount on hand. The amount that is used for an actual honeybee colony is itty bitty. And so working with compounding pharmacies or university pharmacies or things like that, or your veterinarian hopefully can make those available. And again, that's something that we can work with a pharmacy or a veterinarian to help them navigate that. All right, and then you do follow the instructions. I used this photo because this is a classic example of a beekeeper with a margarine container that just says bee on it, applying the antibiotics, because they are very, very difficult to find. Not that the antibiotic itself is difficult to find, it's hard to find a veterinarian who will prescribe them, or if they can prescribe them, to actually get them appropriate. But what you do is you take your hive tool and you can apply it to the tops of the frames, as shown in a couple other photos, and that's done 3 times, 4 to 5 days apart. And obviously you follow all of the instructions on the label. And that's the most common method. Another thing that's on the label is a withdrawal period. All of those antibiotics that are labeled for use in bees are medically important to humans, and so we definitely don't want them going into the food supply. So they can only be used when the hives are not collecting honey for human consumption. So one option, a lot of times it's not a problem because you're using the antibiotic because the colony is so sick. You've got this really weak looking sad hive and it's just not going to be making honey anyway. So that's not a problem. Another thing is to be really careful about spring or fall or, outside of your honey making season. And this is another reason that watchful waiting isn't always the best option because you can say, oh, I have this disease early spring, let's see what happens. And then you let it build up and it starts to get really bad. And now you're in the midst of your honey flow and you have to make a decision about collecting or not collecting honey. We were in that situation with our research hives when we were doing that shook swarm. And we went with this last option where we were only using it with equipment that was not used for honey collection. So these are some of our hives at MSU. We use deep boxes that are painted really pretty for our bees, and then we use media boxes that are white for our human consumption. And so for all the colonies that we were giving antibiotics to, we just added more deep boxes. So they got taller, they were able to have the space needs that they had, but we knew that we weren't going to extract that for sale to humans. Those were just going to be used for overwintering. All right. And there's always the question about using antibiotics for prophylactic use. And I always say it's not prophylactic use, it's metaphylactic use. And then I have to explain what that means. So when we talk about using antibiotics, again, we don't want to be overusing them, but we do have animals that are suffering from a severe bacterial disease that is highly transmissible, and we do want to manage and control that. So when we talk about prophylactic use, that's the case where people are saying you're applying it when you don't see signs of disease, but also you don't confirm that you have it within your operation. Metaphylactic use means you've confirmed that the pathogen is within your herd. So the difference is one of them is really truly kind of preventative. The other one is I know that it's already circulating and I'm managing the disease outcomes knowing that the pathogen's within the herd. So again, the oxytetracycline is labeled for control, which does include metaphylactic use. In the case of like these large high risk operations, so here I love this photo because you can see there's at least 3 or 4 different operations, just judging by the paint colors on the boxes, that are absolutely within flight and drifting distance. This is a very, very high risk scenario in which it's very likely that they're going to get the pathogen. Especially in larger operations, they're not going to have the capacity to do shook swarm every year. And that would be a case where a lot of the control is just going to have to be dealt with with antibiotics until we get better tools. In those cases, a history or a previous diagnosis of European Foulbrood should be sufficient because we know that it's so persistent and because we don't have great cases where we know people can eradicate it. So usually what we see is that people get EFB in their operation and they kind of just have EFB in their operation. And then they have good years and bad years depending on weather and all sorts of things. And so they can choose to use antibiotics based on their past history of having the pathogen in their yards. All right, so by far the best thing that you can do is prevention. Like I said, once you get it in your operation, it is incredibly hard to eradicate. We've had it at MSU, which I kind of love because I use it for teaching and research and all of that. We haven't really tried to eradicate it, but that's not the joy. As Peter mentioned, what we've seen is a huge loss in honey production and in cluster size, and it's not great for colony health. So if you can prevent it, that is by far the most important thing. The ways to do that is practice really good biosecurity. It is at very high rates right now in the US. If you are buying bees and catching swarms and doing cutouts and constantly bringing bees in your yard, at least keep them in a separate yard. We talked about Michigan, but the USDA did just release a prevalence study around the United States, and it is variable by state depending on what's been sent into Beltsville, but Michigan is right in the middle. Like it is pretty normal. So it is high prevalent in lots of other states. And so if you're buying bees every year, especially if you're not doing sufficient varroa control and you're buying packages or you're buying nukes each year, there's a very high risk chance that you're going to get it in your operation. And it's not necessarily going to be the fault of the person that you bought the bees from, because with high rates of inapparent infection, they can easily sell you a colony that looks perfect and has the bacteria in it. And then when it gets into your operation, the conditions are right for it to turn into disease. So that's the big one, is just purchasing bees. And then the other one is being really, really careful about how things spread. So we use nitrile gloves when we're at MSU, and obviously we wash our hands and use alcohol sanitizers. If you use leather gloves, I would do one hive per or one per yard. And again, same thing with like smokers, hive tools, things that are very, very that you're going to touch a lot with your hands or that are going to go into the hive, and clean things often. It wasn't until I started working with veterinarians that I started cleaning hive tools. And now that I think about it, that's pretty gross. And that is pretty common not to kind of clean your beekeeping equipment. So making sure that you're actively cleaning it and removing all of the infectious material from it, or at least all the materials, and then be very deliberate about your equipment movements. You can hear like a newspaper combine here. Just kind of think, if you know you had EFB in your operation, if you're going to move something from yard to yard or this hive to that hive, just know that there's a chance that you could be moving those persistent bacteria even if you don't see signs of disease. All right, so the main points: we don't know a way to completely eradicate EFB from your operation once you have it. The shook swarms definitely can reduce the pathogen load. Antibiotics reduce the time to recovery. So these are ways that we can control the spread. But again, neither of them are actively going to get rid of every single bacteria. And this is a yard level disease. So if you see signs of it in one hive, you can likely find the pathogen in many hives in the yard. So it's something to just be aware of as you manage it. And be really strategic about your equipment replacement and moving things around. Practice good biosecurity. So don't buy bees unless you absolutely have to. All right, so I really like the first question, is if you can recap why EFB is called European Foulbrood and why AFB is called American. And someone pointed out it's funny how high prevalence of EFB we have in the US more than that they have in Europe.
Yeah, so it is a little confusing, like the whole trajectory and the timeline of it. So both of these diseases seem to have been in beekeeping operations everywhere throughout history. There doesn't seem to be any locality to it. The only reason American foul brood is called American foul brood was because Gershom Franklin White, the USDA scientist, was able to grow the bacteria relatively easily and confirm that that bacteria killed the larva. So he was able to fulfill Koch's postulates and determine the cause of the disease first. So the European paper that I'd mentioned described a disease, but it wasn't very specific. And so he termed the other disease that he found European foul brood, which he described after American foul brood. So now it's really just like, when was it discovered and by who? And so he called it European because it was first formally described in Europe. And then I think the question about American foulbrood prevalence, it used to be a huge problem, right? Megan probably knows the history a lot more, but like there was
Yes.
massive eradication campaigns in the United States, and that helped a ton with control. So I don't know if you have any history on that stuff.
Yeah, so well, and I know it mainly from Mike Hansen, who is our old state apiarist. And I believe when they started the program in Michigan, it was about 25% of colonies had AFB, which just sounds bananas to me.
Yeah.
And then they had a pretty serious eradication program where they actually had, right of entry, and they focused really heavily on destruction. And they were able to bring it down quite low. The main difference of why that was so effective with AFB compared to EFB is that the AFB, the bacteria, is not in the adults as much. So it doesn't seem to be as present in the nurse bees. So once you get rid of the equipment, you don't really have to worry about it being in the bees. But when it's in the adult bees, you can't really ever completely get rid of it. All right. So there's another, I'm going to kind of skip around and go in the order. So one question, it says, can a virgin queen from an EFB hive carry the bacteria to a mating nube? So the main transmission that we know about is nurse bees and food. So it seems to be that the larvae get the bacteria through their feeding, and that is somehow transmitted with the nurse bees. I don't know of any studies that show of any sexual transmission or vertical transmission from the queen. However, this is maybe a case of absence of evidence is not evidence of absence. I don't know how well this has been studied. Peter, I don't know if you've come across any queen-related transmission studies.
Yeah. Like there's these old USDA bulletins, so they did tons of research on this and like from from 1912 to like 19 like 40. There was just like a massive amount of research and they were doing all sorts of little tiny studies here and there and like there's claims that like it can be vertically transmitted. But none of these studies are very extensive and they're obviously not recent when you could grow the bacteria reliably. So I'd be super curious if that does play into it at all.
hmm And I just pick up the 1960s honeybee pathology from the library because a lot of these things are really hard to find online too. So even if we know, even if they have been done, they're just kind of sometimes physically hard to access. One of the things for our disinfection study, it sounds like a very simple study, like, oh, you just grow it and then you see what kills it. And Peter can tell you with like his probably years of blood, sweat, and tears trying to grow that bacteria, that it's very, just very difficult to work with. And what we're working with right now is just trying to figure out how to quantify it, so that we can say this thing brings in, this disinfectant results in a drop of this much of the pathogen. But, there's some really, really basic things with just working with the bug that haven't been sorted out that hopefully once we and other people who are working on it get a little further, we can kind of redo some of these studies of looking at some of the basic transmission. Peter, there's a question about if you want to talk about if researchers are using CRISPR to add in different chains or at all working with EFB.
So I don't know, like Megan said, like the bacteria itself is so finicky to grow that not a lot of people are just growing it at all. So there's not been a lot of efforts to genetically manipulate the bug. But I think that there needs to be more research along those lines. We do know it has a CRISPR-Cas9 system. So that's something we've been sequencing a lot of this bacteria, and so we've been looking at the genetics, and there's some interesting stuff there that hopefully we'll be answering some questions with soon. So
It's very interesting to look into CRISPR and phages and things like that. And I think, the paper that we're submitting soon is going to release a lot of sequence data. So there just haven't been very many of the strains in the US that have been sequenced yet. I think that's another bottleneck too, is we just don't have a sense of what's circulating, the regular diversity with it, what it's containing, and as Peter mentioned, like have it hard to grow as well. All right, so there's a few questions I see about the vaccine. They've created a vaccine for AFB, but they have not yet done the same for EFB, which is probably more needed than AFB, which prevalence-wise is true. Is this because EFB is not a spore-forming bacteria and is more complicated? Do you want to do this one, Peter? I have an article to put in the chat too, if you want to start, or I'm happy to take it over.
yeah, I guess I think they're working on the EFB vaccine, but it's been less straightforward. Likely some of the problem is how diverse. I think you saw the kind of genetic diversity that I showed of the pathogen and what exactly you're using. So how the vaccine works is they basically kill the pathogen and they take chunks of it and then it's fed to the queen in this kind of medium. The idea is, and this is kind of like still a subject of ongoing study, is that the queen passes certain fragments of that pathogen onto the eggs in something called vitellogen. And when the larva hatches, they use those pathogen pattern profiles as a sort of a map of the different things it might encounter that might harm it and trigger its immune system. So yeah, the process itself is complicated, and we know very little also just about honeybee immune systems. I found out recently, so insect immunology is also just a kind of a new topic area. I think it would be awesome if we had an option that could even just reduce the burden within a lot of these operations. So I'm hopeful that some of that stuff will pan out, but we don't have any research on it yet for
And
EFB.
we did get copies, or we did get some of the vaccine to trial at MSU this year, but again, it would be next year when we actually get the results from that. But as Peter mentioned, so I just put an article in the chat that is open access, and the title is Lack of Evidence for Transgenerational Immune Priming Against the Honeybee Pathogen Melissococcus plutoneus. And so the honeybee immune system, the way this works, it isn't the same where they have antibodies. So when we test vaccines in humans or other animals, they generally produce an antibody that's really specific. And honeybees just kind of have this immune priming. So you can kind of think of like their immune system is kind of elevated or more activated. That does seem to work for the context of Painobacillus larvae, but it just hasn't seemed to work as well for Melissococcus yet. But so Peter, do you want to talk about the seasonality? So do we believe that the seasonal EFB peaks are due to temperature or honeybee activity or what?
This is a good question.
It
I think
is a good question.
I do feel like this is been observed long enough to where there's probably something there that would explain a lot about this disease, but it just hasn't been extensively studied. I think a lot of people, there's theories that like different food that's blooming throughout the season. Some people have associated it with diet and blueberries because blueberries bloom around a certain time. So maybe like what's blooming at a certain time may provide adequate nutrition for the bacteria to grow and it may trigger it. But nothing's been shown to reliably predict when it's going to come on. But it does seem to be like pretty steady. June is when it peaks, and that was confirmed by USDA's kind of national data too.
Mhm.
So I'd be curious to, it is an area I think we need to look further into.
Yeah, and going back to some of the old studies, that's something that they've talked about for a long time too. So Bailey and Ball, who did a lot of the work in the like 40s and 60s, talked about had some hypotheses for this relating to the demographics within the hive and the feeding of the bees. So, and removal of sick larvae. So there's another question here about hygienic behavior. And Peter and I have published a study showing that we couldn't find an association with colonies that were more highly hygienic that and disease outcomes. That doesn't mean that there, isn't one or it wouldn't be helpful, but there is good data to support that hygienic behavior is really closely linked or is useful for, and this is again what we did the freeze kill assay. And so hygienic behavior as identified by the freeze kill assay is helpful for American fall brood and chalk brood. When we looked into it, we couldn't find that it worked for European fall brood, but the way that Bailey describes it is that if you've got these periods of growth and you've got all of this incoming food, and it changes the amount of food that's given per larva. And when we've got this huge amounts of growth, they are maybe you can have maybe a situation where you have larval neglect where they're not getting as much food because you've got so many larvae in there, and then you also have all this food coming in, so you have bees that are out foraging at much higher rates than being in the hive. And then the other side of that is that they're less likely to do a lot of that housekeeping work and pulling out the disease larva, or it's less important to be removing the disease larva when there is a high food abundance, which is kind of counterintuitive to what a lot of people say is, oh, if you give them a lot of food, then it goes away. And that's one of the things that I hate it when I hear it because that's just not true at all, is that it goes away when there's food, or people be like, oh, a nectar flow will clean it up or feed it. And we have seen so many cases where we see just horrible disease when there's just all sorts of nectar coming in, gallons of diverse high lipid, high protein pollens coming in, and the colonies still get sick. And we've even done a study where we've given them protein, so like protein patties and multiple different types of protein patties and multiple different timings of protein patties, and that doesn't seem to make a difference as well. So it's not just strictly linked to not having sufficient nutrition, but it might be something within the dynamics of larvae and nutrition coming in and who has enough foragers to feed them. Peter, there was a question, that first slide that you showed comparing the 1855 to the another one. Do you think that shows both AFB and EFB co-infection at the same time?
That's a good question. I sort of think that that illustration was a creative illustration from reading the paper because it's clear that they're describing symptoms of both. But if there was AFB in that hive, it would have been pretty easy to pull those spores out of those really decayed larvae because those are really, really persistent in equipment. So I sort of suspect what was happening was that they had a conflated disease that they were seeing out in the field, and they drew illustrations of it conflating both symptoms. And then the actual hives that they were studying when they did their experiments were affected primarily with EFB. So that's kind of my thoughts on it. But yeah, it's a curiosity. We do know there are definitely records of co-infection with both diseases though, so possibility.
Mhm. All right. Does overwintering, like low temperatures or freezing temperatures and brood breaks, help eradicate it? The short answer is no. We see a lot of it show up in the spring. And just to make you feel even worse about that, and I think someone does ask about freezing, we store it in the freezer and then we can grow it after it's been stored in the freezer. So just freezing Melissococcus, as far as we don't know maybe about all the strains, but we know that a large portion, enough strains that allow us to do our research, can be stored in the freezer and reactivate. Is treatment with oxytetracycline essentially the same as the AFB vaccine in that it only masks disease symptoms but allows multiplication anyway? So I'll start, and Peter, maybe you can finish if you want to add. So they're not necessarily the same. They definitely work differently, whereas one activates the bee immune system while the other one actually does prevent the bacteria from replicating. I don't really like the term like masking it because it doesn't necessarily mask it. It does prevent other larvae from getting sick. But because it's something, so even though it doesn't form a spore, you can kind of think about it being more spore-like because it is so persistent. Neither of these things are going to get rid of it from your equipment, or if it's in honey or things like that, it's not going, if you've got places where it's persisting within the hive, giving an antibiotic or giving a vaccine doesn't clean off the equipment. And so, they're both useful tools, or they maybe are both useful tools in terms of preventing, individual animal death and sickness and resulting in better health outcomes, but it's not going to necessarily like eradicate it from your operation.
I would add like if folks remember that side section I showed of the larva that's just like packed with bacteria, this bacteria like basically takes up the entire larval midgut when it's replicating. And when you give the antibiotic, it stops that replication from happening. The bacteria, like Megan said, like, if there's a larva that's full of the bacteria, that bacteria is likely going to, go dormant and persist to a certain extent. How much of it persists, we don't know yet, but it's definitely lowering the pathogen load within the hive. So like when one of those larvae gets infected, if there's no antibiotic, like that's a massive pathogen load that can make its way to other larvae within the hive. So anything you can do to lower that load, I mean, it's the same idea with the shook swarm, is going to be beneficial to the hive and also to other colonies in the yard.
So here's another one. So I know we've started a little bit on the sanitizer or disinfection. Will hand sanitizer eliminate EFB infection on hands?
Uh,
Do you want to describe the strength of the data in the
yeah.
literature of what we know about disinfection?
So there was one paper that came out in like GF White, actually, the guy that discovered it, did a bunch of like assays with like really old stuff, formaldehyde and stuff. Nothing with ethanol. I would suspect we have put the bacteria on different surfaces. One of the summer students we had this year put it on different surfaces and then expose those surfaces to different. This was kind of a preliminary to the larger one we're going to do. It definitely killed stuff, but whether that kills stuff on like on your hands and whether it kills stuff like I would be willing to bet that 70% ethanol and 10% bleach, the stuff that they recommend for decontaminating most stuff, will probably be effective for cleaning most of your equipment, but we just don't have any data yet to say that for sure.
That is something that we are actively working on. Again, our big thing is trying to quantify the bacteria, but hopefully we'll have, and really we're kind of cautious because I know people want the actual recommendations. Like, do I soak it in 10% bleach or do I put the alcohol on for 15 minutes? And that's really kind of the data that we would like to have. I mean, Purell's on your hands is just a good thing anyway. So, again, another question about will long-term freezing of frames 1 year stop the bacteria? One thing I wanted to point, so it won't necessarily kill it all off. Um, One thing that I don't know if we've talked about sufficiently is that we do know that the bacteria can regrow once we put it in the right conditions. What we haven't done a lot of, though, is then seeing if it will also cause disease in larvae, because one of the characteristics of this bacteria is that when you grow it over and over on the different media, sometimes it can lose virulence. And so, just because we can find it or we can make it grow doesn't necessarily mean that it's going to always cause disease in those cases. So, in general, freezing and thawing or using alcohol are good things to rid bacteria in general. So, those are kind of good practices. Maybe it could reduce the pathogen load, but it's not something that we can say, if you do this, you will never have a problem again. All right. Is it viable to treat EFB with feeding syrup and boosting colonies from asymptomatic emerging brood when infection appears new in the colony, essentially diluting the sick nurse bee? So that is a really interesting question. So we know that just feeding is not sufficient, but one of the things that we're hoping to look at is actually these kind of demographics and whether or not you can just add in a lot more nurse bees and kind of change that nursing care per bee, I think is really interesting. Oh, there was a question about does having hygienic bees help keep EFB under control? And as far as we know, no, but I think that's a good thing to do. Oh, here we go. This is the one I was looking for you, Peter. Do you think that it would be possible to test hives in the field with the modified hanging drop method? Do you want to talk about that briefly? And I'll put the link in the chat.
Sure. So unfortunately not in the field. It's kind of a weird method where it requires you, I mean, you'd have to have it set up in your van or something like that to effectively do this out on the road. The basic process for examining this microscopically is you get an intact larva, that it's full of the bacteria. You smash it up and then you take a cover slip. So you don't take an actual glass slide, you take a cover slip, you put some water on it, and you just dilute a little bit of that material in that water. And then you dry it under a heat lamp for 10 minutes. The material will like dry down to that cover slip. And then you have to dye it with carbonyl fusion. So this colors the cells so you can actually see them. And then you basically take a regular slide, you smear oil on it, and then you invert the cover slip so the material's kind of underneath, and then you lower the wet cover slip down onto the oil, and it creates this water oil interface that's really effective for showing AFB spores. So the spores will get trapped within the water oil interface. They'll jiggle around, and it makes it really, really easy to diagnose American foul brood. And European foul brood, the bacteria kind of sticks to the cover slip and forms these clumps of bacteria that kind of look like chia seeds. So it's pretty characteristic once you see pictures of like the bacteria under the microscope. It's pretty easy to diagnose. But yeah, doing it out in the field would be a bit tricky. But like most vets are set up with microscopes, so like I thought like this would be a very useful tool for vets that they could actually have samples. Either you submit a sample or come out and take an actual sample from your hive, and they could actually perform this relatively easy in their clinics.
Yeah, so I put the link in the chat where people can learn how to do the modified hanging drop. So there's a really interesting question about, does Melissococcus plutoniosus exist anywhere else than in honeybees? Is it somewhere in the environment that the bees pick it up? You want to talk about that, Peter?
I would love to, yeah, I really want to like something I do when I'm out in bee yards that have this is I like will like collect material. So we do have like material like hive beetles and like other stuff like, even like nearby water sources, I've collected. But we haven't extensively looked, and I don't think anyone really has. There was one paper that I think found it in native bees or something like that, but it wasn't entirely clear whether that's what they found. So I think it does need to be looked for in other areas, but as far as I know, it doesn't grow anywhere other than honeybee larval midguts, and I think stingless bee larval midguts. Is that the other? I think
There's
it's
a
been seen
couple other species of
in cases
honeybees
where
besides Apis mellifera, but there is a group in, I believe, Washington that is going to start looking for more environmental samples. But I mean, we found it in a forager, but that's kind of the most exciting thing that we found. I kind of was hoping that hive beetles were the problem so we could just like get more angry at hive beetles, but it really does seem to be really specific to honeybees. How far away should a clean yard be from a dirty yard in a boundary system? Ideally, you're going past the flight of the bees, but in reality, what we did at MSU is really just focus on drifting. So where we are is pretty dense with bees, so they're probably never going to be totally remote from all bees everywhere, but at least it's going to be one that is going, they're not going to drift into each other hives. So I'm not going to be able to give you, 0 or whatever, but I think above 2 miles is ideal. You just want to be cognizant of not just 2 miles from your yard, but also to where that puts you in terms of everybody else's yards. Do you requeen with EFB as a management tool similar to the recommendation with managing shock brood? That's another one like freezing and alcohol where like it can't hurt, right? And so that is kind of a standard thing that people talk about. Again, going back to the old literature, you definitely can find places where people talk about how Apis mellifera is resistant to American foulbrood or European foulbrood, and Apis mellifera linguistica is not. Maybe just reverse that. But anyway, there's definitely like people talk about different strains, but that's another one that I don't know of anything really recent where people have looked at host differences. And again, we talked about there isn't really anything recent on vertical transmission. So I think that's an area that definitely needs a lot of study. So I don't know if you want to talk about this. I'm correct in understanding that what used to be called idiopathic brood or snotty brood is now recognized as a form of atypical fall brood.
that's a good question. Uh,
It's-
you know, I haven't been able to when we find atypical and typical in colonies, I haven't noticed any symptomatic difference in the ones that I've looked at. And even in the one, like we had one on campus that was really, really badly diseased, and we screened it and we thought it only had typical. And then, like a year later, out of our isolate, we ended up growing atypical that was actually co-infected at like a lower level. So, like, maybe there's some differences in symptoms with all these different strains infecting the colonies at the same time. But yeah, the snot brood thing, I think that's something other folks are working on that you know of, Megan.
yeah, so that was, I'm trying to think about that. I want to say that was like 2016-ish is that when that term kind of was big or somewhere in there. And the hard part is that people would use the term atypical EFB to also describe when it looked different. So there's typical and atypical in terms of the bacteria, but then in terms of the presentation, and a lot of times what people were talking about was this form of brood disease that wasn't really clearing up. And so, and also that you'd see the cappings getting involved and it didn't look quite the same. It could be different strains because the different strains can kill at different ages, so you kind of see it look different. And then sometimes people were saying that it wasn't testing positive for EFB, but I think in a lot of those cases, what they were doing was sampling the older larvae. So I think it was a lot of what we were calling idiopathic brood disease, or what beekeepers were calling, or snot brood, or the crud was the other name for it. A lot of times I think it was parasitic mite syndrome. That was also when the viruses started to get a lot worse. And I think a lot of times it was EFB, but probably presenting different. Like when you talk to beekeepers who've dealt with it for decades, they say it was kind of different. So, but that is my understanding, is that it is probably mostly EFB. So there are a couple questions about steaming or wax dipping takes that will kill or eradicate EFB. And so they have been shown to get rid of the spores for AFB. Australia has done a lot of work with high temperature, again, times time. And I believe their recommendation is somewhere like 130 degrees Celsius for 20 minutes or something like that. Correct that later because you could find the articles on that. But it is a very, very high temp for a pretty significant amount of time. I don't know that we've actually looked at it for EFB, but again, that's another thing that we can test when we look at the disinfection. And again, people ask about flaming hive tools, but it's the same thing of whether or not you can actually keep it for long enough time to use heat. All right, so we're coming up at the very end of our time. I appreciate all of the people. I'm amazed at how many people have stayed with through the end of talking about 2 hours about European Foulbrood. You are absolutely after our hearts. So I hope this has been useful. I hope to see a lot of you at beekeeping meetings coming up, or online, or training sessions. And I hope everyone and their bees has a great rest of their year.
Bye, everybody.